Hello students, hope you all are doing well. In today's video, we will discuss about the oxidative phosphorylation, which is the last step of the aerobic respiration. In the aerobic respiration, after the glycolysis and the Krebs cycle, our most of the energy is still trapped inside the high energy coenzymes. Those were the NADH2 and the FADH2. Previously, we have studied about the electron transport chain in which these NADH2 and FADH2 get oxidized to produce the electrons. Those two electrons travel through a series of electron carriers that are present inside the inner membrane of the mitochondria. After the electron transport, the electrons are finally accepted by the molecular oxygen to form the water molecule, and as a result of the electron transport chain, many protons are accumulated into the intermembrane space of the mitochondria. So in the process of oxidative phosphorylation, these protons will move through a natural biological motor, that is the oxysomes or the F0 F1 particle, and will phosphorylate the ADP molecule into the ATP molecule, thus creating the ATP molecules. We know as a result of the electron transport chain, many numbers of protons ions are accumulated into the intermembrane space. Whenever the three protons will move through the F0 F1 particles, then there will be the generation of one ATP molecule, which we will discuss in detail in the same video. So oxidative phosphorylation is the process of phosphorylating the ADP molecule into the matrix to form the ATP molecules by the natural biological motor, that is the oxysomes, and this oxidative phosphorylation is achieved by the oxidation of NADH2 and the FADH2 by the process of electron transport chain. So this process is overall called the oxidative phosphorylation. The movement of the protons from the intermembrane space to the matrix of the mitochondria via this biological motor was explained by a scientist, Peter Michel. He termed it as a chemoosmotic hypothesis or the chemoosmotic theory. So let's just start with the mechanism of the oxidative phosphorylation. As I told you earlier in the same video that the most important component for the oxidative phosphorylation are the oxysomes or the F0 F1 particles. These are also called the enzyme complex ATPase. The site of the oxidative phosphorylation is also the inner membrane of the mitochondria, like the electron transport chain. Here in the black marks, that is the 1, 2, 3, 4. Suppose these are our electron carrier complexes, complex 1, 2, 3 and 4. After the complex 4, there is a presence of a natural biological motor, which is also called the F0 F1 particles. It has two components, a F1 head that is embedded inside the matrix and a F0 tail that is present inside the inner membrane of the mitochondria. Our protons are accumulated into the intermembrane space as a result of the electron transport chain. So here in this diagram, we will study in detail the structure of the F0 F1 particles or the oxysomes. This is the inner membrane of mitochondria having our complexes of electron transport chain, like this. Suppose this is complex 1, it will be the ubiquinone. Suppose it is complex 3, there will be the presence of complex 2. Here is the complex 4, and after the complex 4, there are the biological motors are present, which we are showing also here. After the complex 4, there is a presence of these biological motor. It have the two components, a tail component which is called the F0 and the head component which is called the F1. Particularly talking about the tail part, which is embedded into the inner membrane of the mitochondria, it is also divided into three subunits, the A subunit, B subunit and the C subunit. A is a single pipe from which the protons will move into the matrix. Surrounding the A are the C pipes. The C are total 12 in numbers. Surrounding a single A pipe, suppose this is a single A pipe, surrounding it are the 12 C components. The protons will not enter into the C, but the C are surrounding the A, and A is singular in number and the protons will enter from the A component. The B component, which is present in 2 in numbers, the B1 and the B2, made a stator between the head F1 and the F0. It joins the tail and the head. Along with the B component of the tail, the delta component of head is also acts as a stator. Whenever we will talk about the head part, it is divided into the 5 parts, that is the alpha, beta, gamma, epsilon and the delta. The gamma, epsilon and delta subunit is present in single in number. The gamma and the epsilon transmit the torque from the tail F0 to the head F1. The delta subunit forms the stator along with the B subunit of the tail F1. The main rotor part of this biological motor is made by the alpha and beta subunits. Both alpha and beta are present in 3 in numbers. There are the 3 alphas and 3 betas. Total 6 of these components forms the main rotor, which upon rotation of this motor makes the ADP and PI to form the ATP molecules. So this is all about the structure of head F1 and the tail F0. Now we will discuss about the mechanism of the oxidative phosphorylation, that how these ATPs or how this motor will rotate to form the ATP. So we know as a result of the electron transport chain, many number of protons are accumulated inside the intermembrane space of the mitochondria. As the protons are more in number into the intermembrane space as compared to the matrix, so these protons will try to again diffuse into the matrix of the mitochondria. So they won't diffuse simply, they will diffuse by these biological motors which are called the F0, F1 particles. The 3 protons will start entering into the A subunit of the tail F0. Whenever the 3 protons will enter the A will start rotating. Whenever the A will rotate, it is surrounded by the 12 C subunits that we have discussed in the videos also. So the C subunits will also start rotating whenever the protons will enter. So as a result of rotation of the C, a torque is generated which is transmitted to the head F1 by the gamma and the epsilon naught. This torque is enough to rotate the complete motor, and the rotor of the motor is made from the alpha and beta subunits. This torque leads to the rotation of the rotor. The rotor, whenever it will rotate, it will mix the ADP and PI to form the ATP. Thus, entering of the 3 protons forms 1 ATP because 1 rotation accomplished by the entering of the 3 protons. So it is the mechanism of the oxidative phosphorylation, that how these biological motors, the F0, F1 particles, create the 1 ATP molecule. And we know how these protons are accumulated into the intermembrane space because these are the result of the electron transport chain. One NADH2 oxidized to give the electrons and pass on 10 protons to the intermembrane space. One FADH2 transmit 6 protons to the intermembrane space. This we have studied into the electron transport chain. So whenever the 10 protons that are pumped from the matrix of the mitochondria to the intermembrane space try to pump again into the matrix, it will create approximately, because 3 protons create 1 ATP, so the 10 protons will create approximately 3.3 ATP. And the FADH2 create the pumping of 6 protons into the intermembrane space. So whenever the 6 protons will again move back into the matrix, they will lead to the generation of 2 ATP molecules because they will rotate this motor 2 times, hence creating the 2 ATP molecules. Thus finally the protons which are accumulated into the intermembrane space are diffused back into the matrix, creating the ATP molecules, and this is the process of oxidative phosphorylation. So very quickly let's summarize the process of respiration. We have taken the glucose molecule as our respiratory substrate. The glucose is break down into the pyruvic acid by the process of glycolysis. The pyruvic acid, which is produced inside the cytoplasm, moves inside the mitochondria by expenditure of the energy, and that is the ATP. The pyruvic acid produced by the glycolysis process from glucose. The one glucose produced 2 pyruvic acid. The pyruvic acid enter into the matrix of the mitochondria where it go decarboxylation to produce the acetyl coenzyme A. The acetyl coenzyme A undergoes a cycle that is called the Krebs cycle and regenerated into itself. As a result of the Krebs cycle, we get so many FADH2 and NADH2. Note that for one pyruvic acid, there will be one Krebs cycle, and the glucose produce 2 pyruvic acid. So there will be 2 Krebs cycles into the matrix of the mitochondria for one glucose molecule. So as a result of the Krebs cycles, there is the generation of NADH2 and the FADH2 inside the matrix. These NADH2 and FADH2 are oxidized by the process of electron transport chain. As a result of the electron transport chain, many number of protons are accumulated into the intermembrane space, and finally these protons by the process of oxidative phosphorylation leads to the generation of the ATPs. So very quickly we will also summarize the energy chart of the respiration. So here is the energy sheet of respiration. In the glycolysis, a total number of 4 ATPs are produced, but 2 ATPs we have used in the preparatory phase of the glycolysis. So overall energy gain of the ATPs will be 2 ATPs in the glycolysis. A total of 2 NADH2 are produced in the glycolysis. There will be no generation of FADH2 in the glycolysis reaction. Link reaction is the link between cytoplasm and the matrix of the mitochondria. The pyruvic acid that is being produced after the glycolysis enters into the matrix of the mitochondria, where it is decarboxylated to form the acetyl-CoA. The acetyl-CoA undergoes the Krebs cycle. So whenever the pyruvic acid will changes into acetyl-CoA, it will also generate one NADH2. And we know there are two pyruvic acid from the one glucose molecule. So there will be a total 2 NADH2 generation in the link reaction. There is no ATP produced and no FADH2 produced. In the Krebs cycles, for one pyruvic acid, the Krebs cycle occurs once. For two pyruvic acids, the Krebs cycle will totally occurs 2 times. That's why we will just multiply the high energy coenzymes ATP and the NADH2. So in the Krebs cycle, total 2 ATPs are produced in the form of GTP, because there are total 2 Krebs cycles. In the Krebs cycle, in a single Krebs cycle, there is the generation of 3 NADH2. So in the 2 Krebs cycle, there will be the generation of 6 NADH2 molecules and 1 FADH2 in 1 Krebs cycle. So in 2 Krebs cycles, there will be the generation of 2 FADH2 molecules. So if we will total them, a total of 4 ATP, 10 NADH2 and 2 FADH2 are produced. As we know, after the oxidative phosphorylation, that 1 NADH2 is equal to approximately 3.3 ATPs. We generally take it as a 3 ATP, so it will be equal to 30 ATPs. 1 FADH2 is equal to 2 ATPs, so it will be around 4 ATPs. So if we total them, 30 plus 4 plus 4, that is the 38 ATPs. So in the complete process of the respiration, a total of 38 ATPs are obtained from one glucose molecule. But as I told you earlier, the pyruvic acid that is produced after the glycolysis, the pyruvic acid is produced inside the, here it is, the pyruvic acid was produced inside the cytoplasm of the cell. But this pyruvic acid moves to the matrix of the mitochondria, where it is get decarboxylated, that is the link reaction, and undergoes the Krebs cycle. So for pumping of the pyruvic acid into the matrix of mitochondria, 1 ATP molecule is utilized because it is the active transport. And since there are total 2 pyruvic acids, so there will be expenditure of total 2 ATPs to pump the pyruvic acid into the matrix of the mitochondria. Therefore, we use 2 ATP molecules in the pumping of the pyruvic acid. Hence, a overall 36 ATPs are gained. So the total energy gain in the respiration will be 36 ATPs. So here is the all about the respiration process, that how the aerobic respiration is completed in 4 steps, that are the glycolysis, Krebs cycle, electron transport chain, and the oxidative phosphorylation. So that's all for today. Have a nice day.